Assalamualaikum warahmatullahi wabarakatuh, and very good morning to all the audience. Thank you for joining the webinar organized by the IIUM Molecular and Cellular Biology Researcher. I'm Alex. I'm Muhammad Syahdi, the video moderator, and welcome to this webinar series on sustainable natural ecosystem. Let us see and let us comment our This webinar is meant to address the concept of sustainable health, which special reference to the ecosystem. It could be a medium to disseminate relevant and important information in order to develop awareness of sustainable health. Before I hand over the session of the speaker, allow me to introduce the our first speaker for this Dr. Nurhazirah and Azlan. She obtained a degree in Bachelor of Science and Master in Science in Malaysia. She later pursued a PhD study in medical biology in Malaysia. Her research interest is in muscle regeneration study in both culture and animal welfare, as well in aging-related disease. Knowledge, attitude, and practice to animal welfare and consumption integrated in community. She also now working on smart farming to improve the quality of meat of the land by promoting the to all and these you may post your question in the chat box and then speaker will respond to question during the Q&A session. Without further ado, I would like to invite Dr. Nurhazirah to rapidly go over the animal health practice, certain mechanisms and issues.
Okay, Assalamualaikum and good morning. Thank you for the MC. Okay. Okay, so I hope you can see my screen. Okay. So with Bismillah, I will start my presentation. Okay, so my presentation will be on sustainable health and simple issue and issue. Sorry, sustainable health with simple and issue. Okay, so I hope all of you can hear me. Okay. Okay. So what is the definition of the sustainable? Okay, so sustainable comes from two words, which is sustainable and et. Okay, which meaning that it being to sustain, being to able. Okay, and therefore the to sustain meaning that to hold up, to build, to give support to, and keep up. Okay, and what is sustainability then? Okay, sustainable can be defined as something that is able to be sustained. Okay, something that is being bearable or capable being continued to a certain level. Okay, so now this sustainable has been defined in different way because of the social and economic. Okay, but it can define as the process of an action to which human can avoid the depletion of natural resources in order to keep the balance of the ecological system. Okay. So previous, for other university, they have defined the sustainability as the process of living within the limits of available physical, natural, social resource, okay, in which that human are abandoned to thrive in perpetuity. Okay, so meaning that sustainable is we use our natural resources or whatever resources, but without compromising the future generation needs. Okay. So as I said just now, sustainable is not for the environmental, but also for economic and social needs. Okay. Okay, so next, I'm going to discuss a little bit about health. Okay, so WHO define health as the state of complete physical, mental, and social well-being, and not just by defining of absence of presence of disease or morbidity. Okay, so health also comprised in all the five psychological, okay, physiological and also the social state of human individual. Okay, but health also can be defined in relation to our environment and human characteristic in daily life. Okay, so actually health is the fundamental right for all of our human. Okay, not just like without, not just in between race or religion. We do not. It is the our all of our fundamental rights. Okay. Okay, so what about the sustainability? Okay, sustainability, like I said just now, it describes on our environment, our society, and also our economy. So where does health comes into this pillar of sustainable? Okay, so health is actually in between all of these three elements. Okay, okay. So how is it that how is it intercorrelated between health and these three elements? Okay. So first thing, as you know that environment, a good environment provide a good air, a good living environment for us. Okay. However, if there is a bad quality environment, it will lead to exposure to ourselves like pollution, okay, and later on can cause the increase of disease or risk of other disease. Okay. So what about economy? Okay, so they have associated that increase in the economic growth can be seen by the healthy individual because of the productive. Okay, so next one is the association between society and health. Okay, so as you know, the socially active individual live longer. Okay, and there are previous studies that describe the use a poor social environment with social mental problem or even physical health problem. Okay. So just now we talk about the definition of sustainable, and then we talk about definition of health. So what about sustainable health? Okay, sustainable health is the personal commitment to maintain our own health. Okay, so we know that by study, our like government advice is health is to be able to sustain by our balance of our body, our mind, and spirit, and taking care of our diet, physical activities. Okay. However, in order for us to sustain our health, there are certain variables or elements that can interfere with this. Okay. So what are those elements? Okay. So those elements is again our living environment, which is the biodiversity of our environment, and then the climate change. Okay. And also health. Okay, so these three components is the one that control our sustainable health. Okay, so how is it that it can control our sustainable health? Okay, so first thing, we know that there is uncontrollable and illegal harvesting of wild plant species. Okay, so why is it so important or imperative to conserve plant? Because first thing, plant provide the living condition of our own need, which is the air, the water. Okay, and the second thing, plant harvest sunlight, and it is the primary producer of energy in all food chains. Okay, okay. So not only it provide us the things that we need, but also it provide it prevents soil erosion, vegetation. Okay, so this is how the wild plant species are important for us. Okay. However, in sustainable health, okay, there are certain corroded. This wild plant is being corroded or being harvesting. Okay, and therefore it can cause a loss of this plant. Okay, and therefore it can lead to the extinction of this plant. Okay, and therefore the plant conservation is also an urgent task. Okay. Okay, for next issue is the habitat fermentation. Okay, so habitat fermentation is the process in which large expanse of habitat is being transformed into a number of smaller species or smaller area. Okay, so what happened after the habitat fermentation? It can cause the reduce of species abundance and community composition. Okay, so this reduction in size habitat and also increase in border. Okay, as you can see in the figure, it can cause the changes in the composition of the plant and in the animal community, and then later on will cause the change in the biodiversity of animal and also in plant. Okay, so the changes in the land can eventually can expose ourselves and even the human and in animal a new pathogen which later on can cause other type of disease. Okay, so next one. Next one issue that we have facing right now is the decreasing of wildlife population. Okay, it can cause the collapse of all the living things and ecosystem. Okay, so certain the decreasing of wildlife is what we call as the dilution effect. Okay, whereby a greater diversity of intermediate hosts can dilute the pool of hosts, which later on can amplify the transmission and later on can cause the exposure of vector to borne disease. Okay, so there has been proven this previous study that conducted that avian, which is bird. Okay, so when it being fall, the diversity is fall, it can increase the wild Nile virus in US. Okay, and also like Lyme disease and also hantavirus is also increase in falling of mammalian diversity. Okay. And also there have been increase in Chagas disease in Panama and Amazon because of reduced mammalian species diversity. Okay, so next one. The next issue that I'm going to discuss is the imbalanced ecosystem. Okay, so ecosystem can be defined as the state of balance between species coexist with each other. Okay, like us human with animal and plant. Okay, that is all the species ecosystem. However, there are disturbance that can be cause changes in this balance of ecosystem. Okay, it can be by natural or it can be human. Okay. How about ecosystem? So imbalanced ecosystem can cause the ecosystem become fragile, okay? And therefore it can cause a breach between pathogen to human, okay? And this is why the reason of emergence of transmission of novel viruses or even zoonotic pathogen, okay? However, not only for it can cause the emergence of new disease, but it also can cause increase or shift, decrease or increase, okay, a shift in density and distribution of animal reservoir and anthropod region, anthropod vector, okay, as seen in the polar region, okay. Okay. So the next one issue that I'm going to discuss is the extreme climate for animal and plant, okay. So the changes in the heat wave, okay, it can cause the emergence of the infectious disease or allergen. Okay, so climate also influences the distribution of disease-borne vector, which later on can cause illnesses to human. Okay, and other elements or variables such as air or water temperature, precipitation pattern, extreme rainfall event, and seasonal variation also can cause disease transmission. Okay, so not only that it can cause changes in vector or virus, but actually the climate change also threaten our food quality, our food production, and later on we can see it can disrupt in the food prices and distribution system, which later on, like I said just now, it affect the economy. Okay, so not only that climate change can cause changes in the crops, okay, but it also can cause a decrease in the livestock and fish production. So we are actually facing this issue, right? We can see that the chicken production is being low, the meat production also being low, and also we have problem like with potatoes right now. Okay. Okay, the last issue that I'm going to discuss is the conflict between human and wildlife, which is the illegal wildlife trade. So as you know that illegal wildlife trade is it's a long issue. We have debated about this for a long time. So you can see why animal is being poaching for their skin or their bones or ivory. Okay, and then so recent case that we can see that wild animal is being sold as being pet. Okay, so they are in China, in a paper that I read in China, even teenagers have like endangered species for their pet. Okay. So therefore the wildlife trade can cause the threatening to the survival of this wildlife species. Okay, So how can it involve in the wildlife can involve in the transmission of disease is that the wild animal, they act as a reservoir for causal agent, okay, which can stay dormant for a long period. Okay, so the presence of this agent in unusual hosts can sometimes increase the likelihood of error in the process of RNA replication, which later on can lead to mutation. Okay. And therefore, this can cause the increase in new generation of strain or new generation of species that can be very virulent and even resistant to antibiotic or vaccine. Okay. Okay, so all of this issue that I have described just now, okay, later on can create the imbalance between the biodiversity in our climate. Okay, climate change will later on will result in our sustainable health. Okay, you can see the impact is the re-emergence or emergence of a new disease, pandemic, okay, and viral adaptation to extreme environment, the mutation of available strain, and also a more virulent virus. Okay. Okay, so why there are needs to tackle this issue for sustainable health? Okay, so you can see that we have like more populated community. Okay, so when they need a more community population, so means that more habitat will be fragmentation, and then more interaction between animal and human. So these all can be processed, can create a process of a new disease. Okay, so you can see the COVID-19 pandemic and recent flood is part of the causes that can interrupt our biodiversity, our climate change, and for our own health. Okay. Okay, so here are example that I show you. So here are all the diseases that currently we have facing. Okay, so the red color represent the newly emerging disease, and the blue one is actually a re-emerging disease. Okay. Another example that I want to share with you is the Nipah virus. Okay, so Nipah virus started in Malaysia in 1999. Okay, I'm sure all of you are aware of this. Okay, but actually the Nipah virus, although it stopped in Malaysia country, but actually it can be seen throughout in India and Bangladesh. Okay, but the most important notes that we need to take note is that in Malaysia, in Singapore outbreak is through the transmission, the virus is transmission between contact with animal. However, in Bangladesh and India, the virus is transmitted between contaminated dead palm salt and also to human and human transmission. So we can see here the virus is become found a new way to being transmitted. Okay. Okay, so just now that I have discussed about some of the issue that we're facing for our sustainable health, but they are so mostly about I talk about re-emerging and emerging disease, zoonotic transmission. Okay, but there are other issue that we are facing right now because of the interaction in climate change, biodiversity, and health. Okay, we can see that there is a waste management problem. Climate resilience, adaptation, and habitat loss, extinction of endangered species. And then because of the climate change, we can see that we can see that some animals have problem in reproductive. Okay. And another concern that we need to realize right now is that there is a food security is become a concern, okay, a concern issue. So you can see here in the figure in 2019, Malaysia is ranked 28 in food security. Okay, so we are facing the shortage of beef, even in chicken. So we need to raise this issue. Okay, we need to face the issue and we need to come up with solution for this problem. Okay, so So lastly, I want to talk about just a brief discussion about sustainable SDG. So SDG is the international solution to encounter this problem that we are discussing just now. So there are 17 goals. Okay, so they are trying to encounter this problem by certain issue by certain measure. Okay, so we can see here certain 17 SDG. So we need to you can see on the website by your own. Okay, but the most important thing that I would like to rise up is that what about our part in contributing this sustainable health? Okay, so maybe we can create a new smart farming device to help increase in the crops, or even like just now that I have seen is that the changes of waste product into electrical. Okay, so other company like already did the sustainable issue, but what about our part? So we need to consider that. Okay, so that will be all for my presentation. Thank you.
Thank you, Dr. Nurhazirah, for sharing. Now we move on to the second speaker, Associate Professor Dr. Lokman. He is Bachelor of Medical Science and Master of Health Pharmacy from UKM. He completed his PhD in Reproductive and Stem Biology at the University of Sheffield. He is a prominent researcher in stem technology and reproductive endocrinology. Now, I would like to invite Professor Dr. Lokman to share his insight on the use of cellular technology in biodiversity and conservation for the system.
Okay, Assalamualaikum warahmatullahi wabarakatuh. Can you guys hear me? Boleh dengar eh? Hello?
Yes, can, Doctor. But then we couldn't see your presentation slide.
Okay, great. Okay, can you see the slide?
Yes.
Okay. Right. Assalamualaikum and very good afternoon to all of you. Thank you very much for having time for this session, the webinar session. Okay, so I'm going to talk about the use of cellular technology in biodiversity conservation. We have established the frozen zoo in our facility. So natural ecosystem is one part or one focus in our group in IMOLEC. Okay, so we're talking about the biodiversity of wildlife. Okay, natural ecosystem, it is an ecosystem represent the integrations of the biotic community and abiotic environments. All entities play their important roles regardless how small they are. So biodiversity is the variety of different forms of life on Earth, including the different plants, animal, microorganisms, the genes they contain, and the ecosystem they form. So the increase of the variations of ecosystem or organism would influence the productivity and efficiency of the ecosystem. Therefore, the preservation and conservation of endangered species are very crucial to ensure a productive and efficient ecosystem is made, which eventually may affect human health. So there are several strategies on conservation, like what we have in the Department of Wildlife in Malaysia and in each of country in the world. Okay, and then this is governed by the government or non-government organization and also a public. So we can classify into three broad approach, which are in the wild, or we call it in situ, the conservation of ecosystem and natural habitats, and the maintenance and recovery of viable population of species in their natural surrounding. And in the case of domesticated or cultivated species in the surrounding where they have developed their distinctive properties. So we conserve in their own habitat, or we can make a domesticate. We can domesticate them, and then we can use them as a tame animals. Yeah. So we can also have in captivity or ex situ, the conservation of biological diversity outside their natural habitats. Perhaps maybe we put in the zoo or in the wildlife conservation area. And then we have in vitro or germ or somatic cells. This is what we are doing now. So we're collecting the viable biological sample, such as the sperm, ovum, and somatic cell, tissue, and so on. And then this kind of tissue will be banked in the frozen zoo. And then sooner or later, when we develop cellular technology like cloning and so on, we can apply, or we can use the tissue that we deposited in the frozen zoo. Okay, our team effort in conserving the endangered species is more on the in vitro approach, which is more suited on our expertise. And we also work on the collaborative with another NGO and also in Perhilitan to work with the other types of conservation. So when we're talking about the cellular technology approach, currently the scientists all around the world are working on conserving the precious endangered species. Cellular technology approach either the animal already extinct or still in the list of extinction. Okay, so we try to keep as many as their like the cell or the gametes and so on, and then we try to manipulate the sample that we have in order to get back them in the world. Okay, for instance, we have the cell technology, could be in induced pluripotent stem cell and somatic nuclear transfer. If you have a look to this figure, if we can change the cell that we derive from any parts of the body of the animal, like for example, our group managed to get like a cell line from the kidney of rhinos, okay? And then with the technology, we can turn back, or we can produce the pluripotent stem cell from the fibroblast-like cell that we have from the kidney of the rhinos. And when we manage to get the iPS cell, this technology is from the Kyoto University, Japan, so that we can have the other types of cell that we need. Okay, for example, if we need a gamete, so from the iPS cell, we can turn them, or we can differentiate them into the sperm and ovum. Soon we can fertilize both of the gamete, and then we can get the embryo of the real animals, and then we can have the surrogate mom to get the baby of the rhinos. Okay, this is in theory, but we are working on that step by step, you know, because we want to see either is it possible to have this kind of technology using our cell that we got it from the already extinct Sumatran rhinos in Malaysia. But yesterday or last 2 days, we got a good news that the Indonesian managed to get another girls from the Sumatran rhinos, which is a good. This is a natural one, and then that one is a good news for us, and hopefully we can have another more of initiative to get those kind of animal still alive. Okay, and then if we have a look on the another technology of the cellular technology, or we call it SCNT, somatic cells nuclear transfer. So this one is very straightforward if compared to the IPS cell, because IPS cell we need to have the we need to get the stem cell first, okay, pluripotent stem cell first, yeah. But for the somatic cell nuclear transfer, this is like cloning, okay? So we need to only have like an ovums of any species of the, like for example, if you want to do in the Sumatran rhinos, so we can have like ovum from any kind of, any species of rhinos, and then we remove the nucleus of the ovum itself, and then we will take out the nucleus from the fibroblast cell like that we have, and then we add on to the ovums of the E nucleotide, or the ovum without the nucleus of the rhinos itself, and then those kind of ovum can start to grow and become embryo, because the chromosome now is become diploid already. Okay, so when they grow, become embryo, so then we can transfer them to the surrogate mom. Hopefully we can get the baby from this process. Okay, we can do the embryo transfer, and then we can have the possibility to get this kind of baby. So this is the technology, or we call it the cloning technology. And then, so by those two technology, perhaps we can have like, we can bring back the animals. Okay, when we're talking about the frozen zoo, basically the idea of frozen zoo is to ensure we could conserve the animal even though the wildlife is already dead biologically or extinct biologically. So we still have the elements or the life cell, yeah. Because if we only have like a sequence of DNA, or we can have like material, DNA material only, perhaps it's quite far to get back the animal. But if we have like a cell, the living cell, so that's better, I mean, closer for us to get to do the cellular technology. So in cellular technology, the life and dead cell are required for cryopreservation and conducting research related to genetic changes of the animal. Commonly, the sample of wild animal that severely injured will be euthanized after verified by the veterinary officer, or that due to accident will be collected and processed immediately and cryopreserved in -150 degrees Celsius or -80 degrees Celsius freezers. Okay, so this is what we do now. So we will go and collect when the Pahilitan call us. But sometimes somehow we got the sample that already torn away, that already died for several times, so that end up with we cannot get a proper cell line from the tissue. Okay, the meat is already rotten, something like that. So now we try to change our approach, like to get the biopsy from the living animal, maybe from the zoo or from wildlife conservation area and so on. Once we are ready in the future with the advanced cellular technology, it is not impossible to bring our extinct species. Okay, now we're not talking about the future. We are now doing the technology, the cellular technology that I mentioned just now in our lab. Okay, this is our collection. There are various tissue or cell that we collected. So we have of course the Sumatran rhinos of these three Puntung, Kertam and Iman, and from the Malayan tigers, both Javanicus is the bunting, or the bunting is already extinct in Semenanjung, but in Sabah we still have, or we call it Tamadau. This is species of the gore or seladang, yeah? And then we have a Malayan tape, proboscis monkey, pangolin, clouded leopard, and also seladang. Yeah? So this sample potentially could be applied as an attempt for cloning the wild animals. So we have the cell here, and then from that kind of cell, we will try the technology. So like Dr. Hazira said, so this is the interrelated, why we need to conserve the animal, the biodiversity. This is because at the end of the day, it will affect our human health. Okay, this is bring back to the sustainable of health. Okay, biodiversity support human health and society needs, including food security, energy, developments of medicine, pharmaceutical, and clean water. Protecting biodiversity can help us adapt to climate change. Healthy ecosystem will be more resilient and able to maintain the supply of ecosystem services on which our prosperity and well-being played. Okay, and if we not control the climate change and so on, so mitigation and adaptation strategy will positively affect both climate change and environment, thereby positively affect our human health. So this is the key elements. The triangulation here is the key that we need to taking care of, okay, to ensure that sustainable, our health is sustained. Okay, biodiversity, health, and climate change. So in our group, we have the group that looking into the biodiversity, or we call it natural ecosystem. So this is like a diagram showing how, like for example, COVID-19 can cause a very harmful event to our human health. Okay, like we do the vegetation to our land, and then with that, the climate change happen, and it will directly affect the biodiversity, change the polar of the biodiversity, maybe increase in bats, and then decrease in some other types of animal, so that the reservoir in the bat itself will transmit to another endangered species. Okay, and then the human also have like kind of culture to eat endangered species, to eat some like wildlife and so on. So the virus that come from the bats will go to the endangered species or wildlife and then consumed by the human. So human to human interaction will cause more pandemic of COVID-19. And at the end of the day, we are facing with the global health. So this is an example if we unable or we fail to balance our biodiversity. So this is our activity so far. We have our zoo, a mini zoo, like we try to keep the endangered species, like for example, for the porcupines. And then this is the kajang goat that we have. The kajang goat is the domestic goat that being extinct because of we manipulate the genes, we clone with the different species of the species from outside, and then to ensure that they will get a good and healthy in weight, so that this is more in economy. But then at the end of the day, the real species of the kajang goat is getting extinct. So now we try to grow them with good food and so on. We want to see how far the normal or the original kajang goat can go, in the weight, so that we want to see either this kajang goat can be potentially as economic drive. Okay, and then this is like we got like Malayan Tapi that being dissected because of the accident, motor vehicle accident. And then this is our activity for the cell derivations, and then we also grow the food for the kajang goat, which is Napier grass. So with that, thank you very much, and hopefully it will give you some kind of input on that. So Assalamualaikum warahmatullahi wabarakatuh.
Such an eye-opening explanation of the zoo. So We move on to the last speaker for this, Noratikah. Noratikah will a degree in bioinformatics and a master of science on Biodiversity. She also PhD at University of the UK. Her research interests are molecular biology, biology, and metagenomic studies. Her research focus on exploring the complex interaction of microbiology, community, and ecosystem to better understand how micropopulation and ecosystem impact the overhead and development. She will present about the microbiome and the sustainable and the flow is the
Thank you so much, Brother Shahid. So I'm going to share my screen now. So I hope that you can see my screen clearly. Assalamualaikum warahmatullahi wabarakatuh, everyone. Thank you for coming to our webinar today. So today I'm going to talk a little bit about the microbiome and sustainable health. So the outline of my speech today is first, I'm going to introduce you what is metagenomics and also the applications of metagenomics in healthcare. So first, metagenomics is actually defined as the extensive study of various types of genetic material that can be recovered directly from environmental sample, which is further used for many purposes, such as identifications of microbial species, for example, like bacteria, viruses, fungi, and also we also can do a microorganism classification based on this metagenomic study. So
Noratikah, can you full screen?
Was it okay now? Can you see that now? I mean,
it's not full screen, but I think it's okay lah. I mean, yeah.
Oh, nak swap display ni kot. Is it okay now?
Yes, clear. Wonderful.
Okay, now where I stopped just now is about metagenomic and then how the we use this metagenomic as identifications of microbial species such as bacteria, viruses, and also fungi. And at the same time, this metagenomic we use for identifications of microbial species as well, and also the classifications of microorganisms in the environment. So that metagenomics is actually it's very important, as you can just take a sample from, for example, like oral sample or from the soil or from the animal itself. Then you will know what is the microbial community in that particular samples. Okay, so that this metagenomic field, it can not only identify the microbial or microbial species present, but also provide insight into the functional roles and metabolic activities of the microorganism. This happens when we start to sequence the microbiome in that samples, in that particular environmental sample. We do fully sequencing, and then that's how we can get the insight into the functional roles and also metabolic activities of the microorganism. Okay, and then the coupling of this method with function-based activity is a powerful technique for the discovery of new functional genes from uncultured microbes. So so that we don't need to culture our microbes, so that take it just straight away from the samples of from the environment. Yeah? Now, so that this is what metagenomics is all about. And then now I'm going to talk about the applications of metagenomics in health. Okay, here I put three very broad applications of the metagenomics. First is in infectious disease, gut health, and also wildlife conservation. Okay, for the infectious disease, this metagenomics has now been applied to identify an unknown pathogen in outbreak of disease. Okay, an example recently is the COVID-19. And then the shotgun metagenomics can also be used in the discovery and detections of pathogen in clinical samples. In addition to bacteria, pathogens, and viruses, metagenomics has so far seen a little use in the detections of parasitic infections as well, so that it is not just to detect microorganisms such as bacteria, viruses, or fungi. It is also for detections of parasitic infection. And for example, like Plasmodium and Toxoplasma sequences, it was these sequences were found in the metagenome of Egyptian mummies, Plasmodium, Egyptian mummies in 2013. At the same time, if we already have the profile of microbiome in the wildlife, of our food, in the wildlife, or from the food from animal sources such as fishery, poultry, these will be very useful if we face an outbreak in the future, so that we can already identify the source of infection. Okay, an example that I can give you is, we are very familiar with COVID-19. At first, in 2020, when COVID-19 first hit in 2019, end of 2019, we don't know where it's come from. And then in 2020, we started to know that it's come from the bat, right? The host of the virus itself are from bats. And then some sources says that this virus would come from pangolin that was smuggled from Malaysia. So that, and then at the same time, we don't know whether what species of bat as well these viruses come from. Is it the same bat as the SARS, or is it other animals such as MERS, the infection of MERS, which will come from camels, if I'm not mistaken, yeah? And then, so that if you already have a profile of microbiome from the animals or from the food source that we eat, so that this won't be a guessing game during an outbreak. Okay, so I think this is very important for us to look more in details on this metagenomic in health, and then the integrated of zoonotic disease surveillance system is very important to detect disease emergence in human or animal population anywhere in the world at the earliest time possible. So at that time, when the rumors says that it was the COVID-19 come from smuggled pangolin from Malaysia, so that if you already have the profile, the microbiome profile of our wildlife at that time, so that we can straight away eliminate that it is not from pangolin. Okay, so I think, so it is the zoonotic disease surveillance system is very important, and then also to where we can start to eliminate the possibility of the infection at earliest time as possible. And then early detection is also essential to trigger timely disease outbreak in investigation. Yeah, so this has happened as in COVID-19 as well. So I think it is the multidisciplinary teams of professionals that have relevant expertise and also and field experience will identify population at risk and cause risk factor for infection, and then rapidly and widely can disseminate this information so that the immediate and longer term disease prevention and control intervention can be implemented, so that it is not that we have to wait for the samples to be fully sequenced and then go get and then when the outbreak already spread to become a pandemic, and then at that time we only do the swab to take the sample from all of the wildlife and then have to sequence it. So I think so it's going to take a long time if that happened on that particular time, and then so if we already have a profile of microbiome, so that the prevention control intervention can be implemented as soon as possible. Okay, and then also the goal of this intervention will be to control the size and geographic scope of the outbreak, and to minimize morbidity and mortality, and also economic losses in both human and animal population as well. So that this is how we can apply metagenomic in health. Okay, so now I'm going to move to gut health as well. Okay. For gut health, we have this 16S or 18S amplicon sequencing, where we not sequence the samples of all the microbiome from we're not going to fully sequence it, but then we only sequence the amplicon of the 16S or 18S. Okay, because this is why we choose the 16S and 18S, because it is very conserved throughout the species, but then at some region they were a little bit different, so that there we can differentiate between species. Yeah? And then it is a powerful and affordable tool for clinical and also microbiota analysis. It can be used to determine gut microbiome species and their abundance, and also allow to monitor human health and well-being. So that this gut health study were broadly studied overseas. I'm not sure about Malaysia, but then I read few. There's abundance of articles regarding the gut health. To monitor human wealth and also well-being. And then metagenomics sheds the light into development of probiotic as well, so that by monitoring human-associated bacteria communities, and also it is not, we can not only focus on human, but then we can also focus on animals as well, so that by monitoring the human or animal-associated bacteria communities, it will allow us to establish ways to modulate them. Therefore, we can optimize the human health or the animal health or the wildlife health in these methods. Yeah, and then personalized metagenomic has been on the market and is available for purchase as well. So this is not a new thing. Because it has been commercialized, and then it is always better to prevent than to treat. And personal metagenomic is conjunction with personal genomic and epigenomic, maybe the foundations of the healthcare of tomorrow. And also this can be implemented to the endangered species or to our food source as well. Yeah. And then for wildlife, and then my third point is on the wildlife conservation, the microbiome ecology and next generation sequencing offer valuable perspective and tools for investigating and monitoring wildlife health. Because just now we talked about the monitoring human health and well-being, but now on wildlife health, the microbiome community inhabiting animals and plants profoundly affect host health, and also nutrition, physiology, and also the immune system. So that the DNA sequence technology allow us to identify microbiomes among and within hosts, so that the microbial communities is also sensitive, yeah, to changes in the external environment. So that, for example, like we know that we know that we have this in Malaysia, in Peninsular Malaysia, in East Malaysia, the wildlife is so unique to Malaysia because, for example, like to the East Malaysia in Sabah and Sarawak, or because it is from the Borneo, it is very it is an island, so that this animal will be unique in there. For example, like our Sumatran rhinos, we can't find the Sumatran rhinos anywhere. So that because of the changes of the what we call the environment, so that it will change the microbiome in the host as well, so that when it changes, because the microbiome is very sensitive, what happens is it will start to change the metabolic of the animals itself. Yeah, it might because we know that some viruses can cause cancer. So that when these were interrupted, then the microbiome itself was interrupted by, for example, by the temperature. When it is getting very hot, then what happens is this can trigger new disease to the wildlife as well. Yeah. For an example that I can give you, when the bacteria started to secrete their virulent factor, were the Yersinia pestis. Yersinia pestis actually it caused bubonic, it caused plague or Black Death, where this Yersinia pestis, when it was in the flea, and then this flea was actually the host of the flea is actually rat, and then in rat the temperature of the rat was only 26 degrees Celsius, and then what happened when the flea jumped into human, and then the temperature of human is 37 degrees Celsius, and then these Yersinia pestis started to secrete their virulence factors, so that because of the temperature of 37 degrees Celsius that our human body have, so that it's fine in rat, but then it's happened to secrete the virulence factor when it is when the Yersinia pestis were in human, and then what happened? It caused plague to human, and then what happened is half during that time in the 18th century, half of the Europe population were died because of the plague or the Black Death. If you're familiar with this, so that I think the wildlife conservation, the applications of metagenomics in wildlife conservation is very important as well, because the microbiome profile of wildlife can be linked with habitat quality, since this microbiome are sensitive to changes to the external environment. So that I think that will be the end. Actually, that will be the end of my presentation. I hope that you can get you can get some insight on the applications of metagenomics in health, and also if you have any idea or and you need some collaborations on doing this metagenomics, you can come and seek me. I'm here in IMOLEC, and then we can do our research together. I think that's all from me. Thank you very much.
So thank you, Noratikah, for sharing insights and knowledge with regard to the natural ecosystem as a part of sustainable health. Now, we move on to the Q&A session. Question from our chat box. So, the first question is from Dr. Haris. The question is, when the board acting as a conduct for human and prokaryotes. So, are you to answer the question?
Sorry, you are breaking up actually, Brother Shahir. I'm gonna look at the questions by myself. May I know whether both 18S and 16S are conserved both in human and prokaryote. Actually, I don't have the answer for that, but then I think it might be different because of the one is the size of the protein itself, one the RNA itself, one is the size is bigger than the prokaryote one, but I never come to do the like multiple sequence alignment of these two, but then it's a very interesting question. But then I don't think, but from my reading and from I don't think they are conserved between the human and prokaryote.
For your answers. So we move to the next question from Brother Alif, which is Assalamualaikum. I have a question related to the aspect of the Malaysian government towards the idea of frozen zoo. Therefore, I invite Prof. Lokman to answer this question.
Okay, thank you, Brother Shahir. Okay, related the question. So the question is related to acceptance of Malaysian government towards the idea of frozen zoo. Okay, yeah, because frozen zoo is like so-called we rebrand or we name it, you know, because it's like the way like we keep our cell and our cell and our gametes and our embryo and so on in one place, and then we documented well, and so-called we deposited those kind of things in the one place that very safe for longer time, you know. So we name it frozen zoo because big freeze, and then it is still possible to become a living things, yeah. So frozen because of it being freeze, zoo is living thing, so that's why it come the words frozen zoo. And hopefully you're not suspecting that if you go to frozen zoo you can get the animal, the living animal there, yeah. It is only the matter of the materials, the cell, okay, the cell lines that being froze, that have been freezed there. Okay, so in terms of the acceptance of the Malaysian government toward the idea of frozen zoo, because this is the first time that we establish in Malaysia, and then of course in the 2022 budget, Malaysian budget is being listed. I mean, Ministry of Finance already put some budget on that, so that we can apply for that to enhance more of our facility of the frozen zoo. In the other word, government is start to looking into this initiative on conservation of the wildlife. What I can say so, yeah. And then also I've been called to see something related to the frozen zoo with the radio company, Trax FMs, yeah, to talk about that, and then hopefully community will start to aware of the existence of the frozen zoo. Yeah, thank you very much.
Thank you for your answer. So we have the last question. We have another two questions. One question for myself that related to the readiness of our government as well as the law to implement genomic in wildlife conservation. So I don't know.
Daripada sendiri.
Noratikah, are you okay?
Oh, sorry. So that this question is from Prof. Zaleha, right? Will I monitoring? Is that the question?
Actually,
Oh
the
no, sorry,
question is
from you.
myself.
Oh, yeah. Okay, I have a question related to the readiness of our government and local expertise to utilize metagenomics in wildlife conservation. Related to that, we spoke to academic science negara where these were in plan with our government as well. So inshallah, when the grant come in, we will do this very soon, so that I think that our government is looking forward for doing this metagenomic or zoonotic disease surveillance of the wildlife. Yeah, thank you, Shahir, for that question.
Thank you, Dr. Noratikah, for your answer. Right now, we move on to the question from Dr. Zaleha. The question is, will IMORET venture into marine organisms such as horseshoe crab, the living concept, or other alleged species such as shark and corals? So I invite Prof. Dr. Lokman to answer this question.
Okay, to answer of the question of Dr. Zaleha, yeah, we are more than welcome to venture into different types, different kingdom of animals, yeah, because we have animals from the big mammals, and then we have the one from the birds, and now if we have a person, I mean, if we have like a expertise, a group members from the sea, it's more than welcome, you know, because of now we try to have the small fish. Okay, we do in the zebra fish now, and then we also try to do in the latest in sea cucumber, the gamay. Okay, so horseshoe crabs and other like big mammals or big fish in the sea, we are more than welcome to have a look on that. And of course, the way of the derivation of the cells, and then like if you do in this, I mean, using fish or another water animal, I can see it's a little bit easier because of the sterilization terms and anything is not a big matter, because of they didn't need a womb for the congestion, but then they like lay an egg, and then with the environments of the water, so it's even a bit easier to do that actually. Yeah.
Thank you, Prof. Lokman, for your answer. Is there any question?
I have a question to Prof. Lokman.
Yes,
please,
yeah,
Dr.
Dr.
Azlan.
Ahmad, yeah.
Yes, Prof. Lokman, thank you for your presentation. And my question is on the security of the frozen zoo that is established at IIUM. How good and secure the samples are at our frozen zoo?
Yeah, we have the system, and then to be honest, we are towards having the working with the Prof Hafiz toward having the ISO, ISO certification, yes, certification, and then we need to have a proper system because of previously I've been called for meeting of the engaging our local, what we call it, a tropics country, okay, Malaysian, Indonesian, Thailand, Singapore, okay, all the tropic country will merge together on having like cell center of the endangered species. So this is that's a good question when we say because of the security, because every country have their own like so-called belonging. So that like in Sumatran rhinos, Indonesians say, okay, they still have the Sumatran rhinos in physical. So they like they want to keep the cell or the animals as their own. Yeah, so that's why we have to have a proper system to ensure, or maybe like if the different country want to deposit their cell into the frozen zoo that available in Malaysia, so we need to have a proper like MOU and proper licensing and proper date of birth of the cell and so on. And then with that, so the cell, we know that the cell is supposed to be used by any purpose and not to be used if without any permission, something like that. So this is the process that actually we are trying to develop in order to ensure that the security of all the cell or of all the gametes that are being deposited in the cell in the frozen zoo, it is safe lah, something like that.
Prof, I would like to also add some more information regarding the question that is raised by Prof. Dr. Zaliha. Regarding the horseshoe crab, the complete tissue samples collected from all the 4 species of horseshoe crab has been collected, and in collaboration with the National University of Singapore, I have already transferred all the samples to NUS, and it is stored at the Lee Chong-Wei Museum at NUS. So this is a comprehensive collection for more than like 2,500 samples, plus having the sample, all the 4 species from all over the world, including the samples from the United States, supported by the US Geological Survey and many other institutes. So the second batch of the sample that is actually with us, so we can preserve it in our frozen zoo in the near future in Aimale, inshallah.
Yeah, inshallah, no problem. So do you have the cell line of the horseshoe crab?
No, I proposed this concept like few years back, and now we have only the tissue as well as the blood samples from the crabs.
Okay, we can add on the cell line. We can do that with.
So it's about 2,500 to 3,000 samples, Prof.
Wow, that's cool.
Fantastic. Another thing that I wanted to ask, why don't the Frozen Zoo, in collaboration with, can have a collaboration with some of the established repositories like Lee Chong-Wei Museum or something, so they have free access to their system, so we can preserve the samples, and by the library system we can even access the deposited samples through the loan. Prof.
That's the idea. Actually, that's the idea. The meeting that we have that I have attended. Actually, that's a chat by the Singaporean representative. So we want to get like one words, between all the countries, so that how we can go about, and then we being monitored by the European and American supervisor, from the San Diego Zoo, actually, so that they monitored us for the tropics area, so that we will get some like one MOU related to that, so that when we have those kind of things, so maybe what you say will be materialized, inshallah.
Inshallah, that's great.
Okay.
Okay, thank you to the speaker for the Q&A session, and for the attendees that asked the question for this session. It was a pleasure to have you with us. You can now invite all the participants to switch on your camera for conversation.
Gambar satu.
So we will take the photo in counter of 3. So you can open your camera. So we will not take the photo. Okay, thank you for the so this was the mask. Thank you for the attendees. We hope you have very valuable information and the system in the to get the not organized by the let's end our session as well. Yeah.